Good morning, everyone. So we are continuing today with the genomics module. So today's session is the third session of this module and is called Comparative Genomics. So you have here the learning objectives for today's session. So we will be seeing today what comparative genomics is all about in a first introductory part. And we will be throughout the whole session, taking a tour together to what comparative genomics is all about. We will start by asking very basic questions in order to understand the genomic variation and then the comparative genomics. Then we will be seeing the input that is needed for these analyses, the methods, and then the output. So today we won't be seeing different videos for each of these parts. We will be going throughout the whole session and I will be highlighting the slides where the different sections are, but the first introductory part would be on a separate video. So here are the bullet points for your learning objectives for today's session. So we will navigate through one mainly of the genomic resources that are available today, and you will be trying to focus on a specific gene in order to retrieve different kind of informations, the sequence, for example, the variants that are annotated for that gene, try to retrieve orthologs, information on sequence conservation also, etc. And we will also be viewing and interpreting genomic alignments. So let's start with what is comparative genomics, so a very basic introduction to the definitions related to comparative genomics. So comparative genomics is based on the fact that a certain level of genomic variation is happening in all organisms, and these changes would affect several features of that genome in terms of structure, of organization, of functions that are encoded in this genome, and these changes would help, or monitoring these changes would help also to monitor the evolution between organisms. So comparative genomics deals with the process of comparing whole genomes or parts of these genomes in terms of their sequence. So the goal is in fact to identify the similarities and differences between features in these genomes and identify the evolutionary relationship between these organisms. So basically you would be comparing whole genomes or parts of genomes. So when we say parts, these are big chunks of DNA or even smaller portions like genes. And what you will get is two keywords here, so features to compare in a genome, and you'll also get an idea about the evolutionary relationship. So as I told you, two keywords here, features and evolution. So doing comparative genomics is basically taking different organisms or different genomes of these organisms or different features, like genes for example, of these organisms, and then try to compare them. So you could be doing different kind of things. You could be trying to compare organisms that are relatively close to each other in terms of evolutionary relationship, and then you would get out of this comparison a certain level of information. But you can also be trying to compare a completely distant organism, and you would get completely different information out of it. So I very much like, usually when I do some courses, to highlight complex ideas using very, very basic examples. And I will be taking here, I would say, a very local, simple example, and we will be seeing how it is to compare dishes instead of organisms. So imagine for each of these organisms, we would be having dishes, and I'm taking, as I told you, a very local example. I very much love the Mediterranean cuisine and the cuisine we have here in Tunis, so I decided to take the example of the couscous. So imagine you would be comparing this one to the second one, and then you would be trying also to compare it to a completely different one which would be this one. Okay, so you can start thinking about the differences on the different features, in fact, that exist between these different organisms, which are represented here by the dishes. So the differences between the different dishes. So you can start think about these differences and what kind of information you can extract out of these comparisons. And in fact, I will be stopping here this first introductory part. And you can start think of what is there to compare. And then after taking the whole class today, I hope you will be able to better understand these differences and what information you can get out of it. And we will be coming back to these differences and highlight what interesting information there is to get out of this. So this is a very basic example to take, and that explains what comparative genomics is all about. So I'll see you for the next video.